It's the top of the hour, so we're going to go ahead and start. Welcome to today's Arctic Research Seminar hosted by the Arctic Research Consortium of the United States, or ARCUS. Today's team includes Stacy Stout, Kubajenda, and myself, Betsy Turner-Bogren. Our seminar, entitled Evidence for Massive and Recurrent Toxic Blooms of Alexandrium catenella in the Alaskan Arctic, will be presented by Don Anderson from the Woods Hole Oceanographic Institution. A link to an online survey will be available at the end of today's webinar, and we'd appreciate your feedback as well as any suggestions for future webinars. We have a couple housekeeping items before presentation begins. You have two viewing options today, gallery view and speaker view. Gallery view allows you to see the webcams of other participants online. Speaker view allows you to view only the presenter's webcam and presentation. To switch between those views, use the button in the upper right-hand corner, which is indicated by the red arrow in the screen. Zoom controls, including chat, can be found by hovering your mouse over the bottom of the Zoom window. You are invited to chat with other webinar participants at any time. To do that, click the chat icon at the bottom of the screen and select the name of the participant with whom you wish to chat. There's also an option to chat with everyone participating in the webinar. Questions to our speaker will be addressed during the Q&A session following his presentation. If you're joining by Zoom, please type your questions into the chat window at any time. I'll record those in the transcript and read them aloud during the Q&A after his presentation. If you are joining by phone, you will need to raise your hand by pressing *9 and unmute yourself by pressing *6 to ask your question aloud. I will give you a reminder of these guidelines just before we start the Q&A. For any technical problems today, please contact Stacy Stout or Kubajenda via email. Stacy is at Stacy, S-T-A-C-E-Y @arcus.org, or Kuba, K-U-B-A @arcus.org. Before we begin, we would like to thank the National Science Foundation Office of Polar Programs for their financial support to ARCUS and to this seminar series. Now I'd like to welcome our speaker. Don Anderson is a senior scientist in the biology department at the Woods Hole Oceanographic Institution. He presently serves as director of the Cooperative Institute for North Atlantic Region and as director of the U.S. National Office for Harmful Algal Blooms. His research focus is on harmful algal blooms, or HABs, commonly called red tides, covering topics ranging from molecular and physiological studies of growth, sexuality, and toxin production to the large-scale oceanography and ecology of HABs, including numerical modeling, forecasting, and a range of monitoring and management strategies, many reliant on novel instrumentation and biosensors. A current research focus is on HABs in the Arctic. Anderson is author, co-author, or editor of over 330 scientific papers and 14 books. Please join me in welcoming our speaker, Don Anderson. Don, take it away.
Great, thank you, Betsy, and thank you for the opportunity to talk about something that's very exciting and very interesting in my science world. I am actually one slide too far. The first thing is to acknowledge all of the co-authors that have been part of this this study. You can see this long list of people in many different institutions, but I want to have a special recognition or shout out to Bob Picard, who's been a co-PI and lead chief scientist on a number of the cruises that we will be talking about, and Evie Fashon and Pagan Lin, who have been really helpful in a lot of the data analysis, but lots of other contributors as well. So the first thing to do is to talk about harmful algal blooms, or HABs, or HABs as we're calling them. Some of you know about these and don't need this, but for those who don't, these are a global problem. They're increasing in frequency and geographic extent in many regions, and I hope I can convince you this is now including the Alaskan Arctic. There's many different negative impacts, public health threats of many different types, ecosystem damage, and of course, a lot of economic losses. Today, though, the main focus will be on two of those impacts, what we would call two of the human poisoning syndromes that occur when humans or animals eat toxic shellfish, occasionally fish, where those animals have accumulated these toxins during their feeding. Like filter feeding shellfish will accumulate toxins from the algae that they're eating. One we call PSP, or paralytic shellfish poisoning. Another one called amnesic shellfish poisoning, excuse me, or ASP. And when that particular toxin from ASP moves through the food web to animals, not necessarily through shellfish at all, we call it domoic acid poisoning, or DAP. And so those are the ones that are the most relevant to the story, that region of the Arctic that we're going to talk about. And I guarantee you we will talk about that region, but I'm actually going to start in the Gulf of Maine, where I've been working for decades on the problem, the PSP problem caused by this dinoflagellate Alexandrium catenella. And I hope you'll see later why I'm making this little deviation, because I think it really gives you some context within which to view the new results that we're getting up in the Arctic. So as you can see, the Gulf of Maine has these major current systems, including this coastal current, the Gulf of Maine coastal current that travels down the coast. And as I said, the organism is the same. This is Alexandrium catenella. It's a dinoflagellate. You see two cells here. They're microscopic. They are typically reproducing through what we call asexual reproduction, or just binary fission. Two cells make 4, and 4 make 8, and so forth, until you get what we call a bloom in the surface waters. But at times, those cells will go through a sexual process where they form gametes that fuse together to make a zygote. And that produces this resting stage, or cyst, that you see here on the left. These are resistant cells that can stay in the sediments for much of the year, then germinating to initiate blooms, and then the blooms drop new cysts to sort of start that cycle all over again. So in any study of Alexandrium, it's obviously very important to learn how many cysts there are and where are they. And in the Gulf of Maine, over the years, we've mapped out the cysts on many occasions. This is a composite map. And what you see are two major cyst seed beds, or just cyst beds, if you will. One up here in the Bay of Fundy, and one down here in the Western Maine region. You see an area in between where there are not many cysts at all, areas offshore, again, where there are not many, and areas down here in the south where there are not many. And with many years of work, we've come up with both a conceptual model that you see here, and in the inset, a numerical model of Alexandrium population dynamics. So let me walk you through it very quickly. As I said, there's two major cyst seed beds here in the dashed lines. And the blooms that occur are very highly seasonal. They occur starting in April or so, but in the wintertime, these cells are gone. They're down in the mud. And what you have is a strong coastal current that moves down along the coast this way. So that, as you can see in this animation, as the cysts germinate, they are being carried as vegetative cells down along the coast, where they grow and divide. And so what you have then are parts of the coast where the bloom is associated with the germination of cysts, but other parts of the coast, like down here in Massachusetts, where everything is advected, everything is carried down to us from blooms that were established to the north. And what you also have in several areas, obviously these seedbed sites are areas where year after year after year, the cysts are being deposited after blooms, so they then start the blooms the next year. So you have annually recurrent blooms due to the new deposition of cysts in these same accumulation zones year after year. So we see considerable interannual variability in the abundance of the cysts, but very consistent and persistent cyst bed locations. All right, so now we've been working in the Gulf of Maine for a long time, but my eye has always wandered up to other regions of the world. This is a map showing where the PSP problem occurs around the world. You can see how widespread it is. But if you look up into the colder waters, you see a dot or two up along the coast of Greenland and maybe Iceland, but really nothing much known above the Alaskan Arctic or above the Russia. And so over the years, I've been trying to get into those waters to see what we could find. And the first opportunity came working with colleagues at the Alfred Wegener Institute with two cruises, one in 2012 and one in 2017. all along the west coast of Greenland. And you can see in the first cruise, we started at the tip and went up to a little bit above Disco Bay. The second one started a little higher at Nook and then went up to about 74 degrees north. And part of the cruise was in fact looking for Alexandrium and trying to see, was it there, how abundant was it, and just what kind of environment is it in. And I can summarize all those cruises here by just saying that only a very few Alexandrium catenella cells were observed in surface waters across all the stations in both of those cruises. They were both in August, a great time for these blooms. This is both in fjords and coastal waters. We found cysts in many, many stations, including as far north as we went, but the concentrations were extremely low, roughly 100 cysts per cubic centimeter. You'll see a little later that that is a truly low number. Now, the sediment temperature was consistently right around 0 and the surface water temperature 2 to 4. And basically, again, I'll show you more results as to why I'm saying this. The Greenland coast simply does not currently support large blooms of Alexandrium catenella right now. I just think it's too cold. We do see low abundances of cells. They're able to persist along the coast, particularly in some of these restricted fjord systems like around Disco Bay. But basically, there's northerly transport in that Greenland current that distributes the species and it cysts broadly, though at very low numbers. So that was a good result and not very exciting, but still useful. So then my eye, it has been turning for a long time to the Alaskan region, and I had written, I don't know how many proposals, 2-3 proposals to NSF saying, let me go up there and look. Let's see what's going on in those waters. Is this organism there? How abundant is it? And I kept getting declined because people say there's no problem there. We know there's a big PSP problem here in Southeast Alaska along the Aleutians and maybe starting out into the Pribilofs or such, but there's no problem there. And so why should we give you money? But then a number of things changed that picture. It was a groundbreaking paper, a frustrating one to read because somebody did what I was hoping to do. This is Natsuki et al. In 2017. A Japanese group went out, and this shows you the Bering Strait, and right north of there in this Ledyard Bay, the Chukchi Sea region, this huge circle shows you how many cysts you have per cubic centimeter of Alexandrium catenella in those waters. They only looked at a handful of stations, but at this one, they found over 10,000 cysts per cubic centimeter. Remember, the Greenland numbers were around 100. So they've found a lot of cysts there. And then in another study, this is a little harder to see, but you can, if you look at the size of the circles, now they're looking at the vegetative cells, the cells that are in the surface waters. And this is a cruise in July, another in August, another then late in the year in September. And what this showed was that there were populations entering into the Chukchi Sea area, but they were being transported from the south, right through the Bering Strait, right up in what they called Pacific summer water. And you can see that in July and also here in August, what is clearly a transport phenomenon. So they were seeing a large number of cysts here, but the cells they were seeing were associated with an advected population from the south. So then how do these cysts get there, and why are they participating? Again, they couldn't answer that question. And to just put a little bit of this in context, here are some of the major current systems that you'll hear me talk about. This Bering summer water here in the center is roughly equivalent to that Pacific summer water that they were referring to as the pathway for the cells they were observing. Bob Picard and I have been focusing much more on this Alaska coastal current, Alaska coastal water. And you can also see over here this water that comes up from the coast of Russia, the Anadir current, which I think is also very important in this process. So another event that happened was this publication, this paper by my colleague Kathy LaFay in 2016. What you see here is a map showing the detection of the two toxins, domoic acid, that's the ASP toxin, and saxitoxin, that's the PSP toxin, in 13 different species of either stranded or harvested marine mammals. So all different foraging strategies, all different sizes, all different habitats that they're in. And you can see that throughout the region, it's no surprise to us in the HAB community that down in the Gulf of Alaska, you have both toxins being detected in many of these animals. But it was quite noteworthy that all the way up, including into the Chukchi and the Beaufort Sea, these toxins are showing up in the tissues or in these animals in such a variety of animals. So clearly toxins are there. It's not just a problem that I'm trying to make up to write a proposal. It's there, it's already entering the food web. And then a number of other events kept happening. There were walrus mortalities, like 39 walruses died along the Bering Strait, and at least a few of them were tested, and one of them had very high levels of saxitoxin in them. And concurrently, there have been millions of seabirds dying. And again, no absolute proof that they were dying from some of these toxins, but people were asking questions. Could the toxins be involved, or is it starvation or something else? So finally, we were able to get funding both from the NOAA Arctic Research Program and from the NSF Polar Programs. This just shows an example series of transects for that's representative of a number of the cruises that you will see data from. And many of these were on the Healy or other vessels. A lot of different types of data were taken, but I'm going to talk mostly about just some surface water samples and some sediment samples looking for the cysts. So to remind you, this is the life history. We're going to be looking first at the cyst, and later I'm going to turn and look at what's up in the water column. So here's the first bit of data, this one from 2018 and 2019. In this case, we took cruises from 2 August cruises in 2018 and 19 and have combined them. This shows the cyst distribution in bottom sediments. And you can see the numbers here, cysts per cubic centimeter with the bright red colors, meaning 10,000, or even the darker colors up to 20,000 cysts per cubic centimeter. And what we did by combining these two, the purpose was to give us a true look at the real spatial extent of the Alexandrium cyst distribution, and also a general levels of cyst abundance. Where we had overlapping transects, we averaged the counts. And so I see a number of things here. The first is a massive Alexandrium catenella cyst bed. It extends 150 to 200 miles offshore at least. Notice we couldn't sample further, and our last stations had detectable cysts, and therefore, without doubt, the feature extends into Russian waters where we couldn't sample. It also extends hundreds of kilometers along the coast. You see in the Beaufort Sea region that there are no cysts. There are low numbers of cysts, detectable but very low. You see the same actually in the northern Bering Sea. Whereas in this region, we saw up to 17,000 cysts per cubic centimeter. And that, except for one other report, that's the highest ever reported anywhere in the world. Many times higher than we see in the Gulf of Maine. The only other higher count is just off to the west a little bit here in Russian waters, not far away. So we're seeing these very high numbers in a large, expansive cyst seedbed. Now, another very interesting feature in the 2019 cruise, we actually picked up what we call a second cyst seedbed right off of Utqiagvik or Point Barrow. And the first cruise, we didn't see it because we didn't have transects over there, but you can also see then there were large numbers of cysts, 14,000 cysts per cubic centimeter up there. So you can sort of see where I'm driving at a little bit with this Gulf of Maine thing in the beginning. What do we see here? We see two major cyst seedbeds and a major current system, and I'll come back to more of that later. So there's the cyst distribution. How does it compare to what we've seen in the Gulf of Maine? Well, in this case, with using this equal area projection, so the area is exactly at the same scale. The cyst abundance is plotted with the same color and contouring. And so you see that this Gulf of Maine feature that I've worked on for years, and that for the longest time was the largest of these anywhere in the world that's been documented, it really looks rather wimpy in comparison. It's Basically, the one here in Alaska is 5 times larger, and it contains over 6.5 times more cysts. And for context, recognize that for decades and probably longer, many decades, the Gulf of Maine cyst seabeds has basically supported large-scale, annually recurrent blooms year after year after year. And now we have described this feature out here that's many times bigger and more dense. Okay, so among the questions that we are trying to address is why are these cyst seabeds located where they are, and why are they so large and dense? And one of the ways that we can just say, well, maybe the blooms are big and it's an ideal depositional environment. I'll talk a little more about what that might mean. But there's another wonderful idea that I borrowed from this paper by Paul Wassman, where they talk about the trails of life and death. And there's something called the trail of death hypothesis, dealing with like zooplankton that can be washed into the Arctic, but they cannot complete their life cycle, their life history, because it's too cold. So in that context, we can hypothesize that the blooms that are being advected from the south, as reported by the Natsuki paper, they're being transported in these relatively warm surface waters. They could then form cysts that fall into bottom sediments where the temperatures are too cold to support significant germination the following year. So in theory, your cyst numbers would just accumulate through time, because and get maybe to these huge numbers that we're measuring, basically due to the imbalance between the inputs and the losses. So in this case, we also should point out that these cysts are very resistant. They can survive from 10 years, 20 years, even 100 years in some reports. So what we would have then is what we have termed in a common way a sleeping giant, an Arctic cyst bed that would represent a significant and dangerous site for in situ bloom inoculation as the waters warm. So this was the basis of one of those proposals that got rejected. I couldn't believe it. It's such a good idea. But anyway, we finally got funded. So let's look back, and we'll come back to that. Let's take the vegetative cells now. Now this is kind of small, and I'll zoom in. We'll get more detail on this in a second. But if you look at the 2018 and 2019 cruises and the blooms, now we're up here in the surface waters. And so what we saw were dense blooms of these vegetative cells in both 2018 and 2019 in August. We took a November cruise in 2018 and found virtually none of these cells throughout the region. So that really makes it clear how seasonal they are. Both of these blooms, 2018 and the two panels down here, 2019, had dangerously high cell densities. I'll explain a little more what that means in a second. But during both years, we had very, very few cells south of the Bering Strait. So let's look much more closely then at 2018 first. And what you see, and you can imagine our excitement when the vegetative cell map was produced and it's sitting directly over that huge cyst seedbed. It was like, wow, this is amazing. We should write a paper instantly, because it looks like that huge cyst seedbed is functioning as a site for local germination and bloom development. And you can see that it extended out again as far as we could go, and probably further into these unsampled waters. And so the concentrations that we observed were the highest concentrations, as you see in this TS diagram over here. These brighter colors are associated with the Alaskan coastal water, and that it's not in the Bering summer water, which would be this. We didn't get a lot of samples from there, but it is definitely part of the Alaskan coastal water, which is the warmest and the freshest type of Pacific water. And the maximum concentrations that you're seeing here are in the 5,000 cells or higher. And that's easily an order of magnitude above levels that are known to be dangerous in other areas, and including in the Gulf of Alaska. So now let's look at 2019. And then you say, whoa, I'm glad I didn't write that paper, because Now the situation's gotten much more complicated. And we had a real fortunate situation where one cruise, you can see, went from the 4th of August to the 19th, and a totally separate cruise went from 20 August to September 11. So they were partly overlapping in their time, but also more, they were overlapping in their coverage. And you'll see why that's important in a second. So what do we see in August is just the same sort of thing that Natsuiki saw in his study, which is a population just north of the Bering Strait. And as I'll talk about a little bit, a number of cells up here, but the large number of cells are here, clearly indicative of an advected population. And these cells, what are they associated with over here, but with this Bering summer water, right? So that's interesting and consistent with what Natsuiki saw, but not at all consistent with what we saw in 2018. But several weeks later in this IERP cruise, what we saw then was a bloom up here near Icy Cape. And this case, the highest cell densities are associated with the Alaskan coastal water. And the thought was, and Bob Picard believes, that this Bering summer water has veered off towards the west, towards Herald Canyon. We couldn't sample there, so we don't know. But notice, as I mentioned, that we did pick up a patch of cells up here near the Barrow Cyst seedbed, which was, again, that's the second location where you might expect some sort of local growth. So if I look at these together, between 2018 and 2019 that you see here, we see two bloom mechanisms. The first one, the transport of established populations from the south, as has been hypothesized by others. But we also see evidence that we could argue is localized in situ cyst germination from both the Ledyard Bay and the Point Barrow cyst seabed. So the question, a number of questions are arising, which of these mechanisms is valid, or are both of them? So how do we go at that? Well, the first somewhat related question is, let's try to explain why that seedbed is where it is. And Bob Picard and Pagan took this composite data set from the shipboard ADCP records from 45 different cruises, largely in the summer, and created this depth average velocity map, where the colors and the vectors here show you how strong the currents are. And basically what it shows you, here's the Bering Strait, the water really roaring through there, and you see a lot of high velocity over here. But what it shows you is that the flow is weak in this Ledyard Bay area where our cyst seedbed is. And you therefore, it would be conducive to the settling of material from the water column to those sediments. And there's a lot less data over here, but on this side of Point Barrow, velocities do slow down. And that's also, many of you probably know, it's a well-known location of a bowhead whale hotspot. And that's a recurrent feature where there's lots of dense aggregations of zooplankton and fronts and so forth, and a lot of dinoflagellate blooms, like frontal systems and low velocity zones. And so it's logical this would also be a deposition area for cysts. But again, what's the source of those cells and the cysts? Are they from which of the two mechanisms is functioning here? So let's go back to 2019 again, and these two separate cruises that I said we're very fortunate for us. So these suggest that both mechanisms are at work. So the first observation, as I showed you, really does look like an advected population right through Bering Strait. Whereas this one, a few weeks later, we believe is much more representative of local cyst germination and is perhaps being augmented with some vegetative cells. Now, why do we say that? Well, first, as I said, this population associated with Bering summer water, this one with the Alaskan coastal water, right? That's one bit of evidence. And again, Bob thinks that the Bering summer water sort of veered off to the west. But if you do the calculations of velocities and say, let's put a particle here how long will it take to get up towards Icy Cape where you could have this type of transect? And Bob and Pagan tell me that that it should take much longer for a parcel of water to advect from Point Hope to Icy Cape, so roughly 50 days, than the time between these two transects at roughly 25 days. And then you add to it, there's a signature up here that I mentioned before with this aqua color, where it does look like there are blooms already developing up near the near Point Barrow and Icy Cape, well before this other cruise. So smoking gun, yes, but certainly not convincing. Just we think both mechanisms could be operated. So how can we take it further? And one way is to look at temperature and germination and temperature and growth of Alexandrium. So first of all, looking at the temperature pattern in recent years, this shows you 5-year bins of average temperatures of bottom waters in July and August and September in the Ledyard Bay and the Barrow cyst bed. So the red is Ledyard Bay, the blue is Barrow. And you can see the number of profiles up there. And what you see is that things have warmed in the bottom waters. And if you go back to let's say 2000 or even 2004 to 2008, roughly a 2.5 degree warming of bottom waters in Ledyard Bay and a little less than 2 degrees in the Barrow region. Okay, well, you've all heard things are warming, but what does that mean in terms of Alexandrium? Well, there I've got to take a little short diversion into the physiology of these cysts. This is some data generated by a former student, Alexis Fisher, and it's really very revealing. What it shows is the days to germination versus temperature of Alexandrium from all of these different places: Chukchi Sea, the Western Gulf of Maine, Eastern Gulf of Maine, and Nauset Marsh on Cape Cod. And so we've taken sediments from all of these areas, treated them a certain way, so you have what we call quiescent cysts. They're ready to germinate. And then you expose them to different temperatures and say, how long until, let's say, 50% of them germinate? So as you can imagine, if it's warm at 12, 14 degrees, it may take only 7 or 8 days. But if it's really cold, it can take several months for those cysts to germinate. And what we've found, and this is very, very comforting because it's very similar to what we see in the higher plant literature, is that there is a degree day functionality that collapses all of the data from these different regions together. You know, degree days, you hear about them as how cold a winter was it, or how many days do we wait until we have to spray our fruit trees to get since the insects are dormant and are emerging. And it's a metric that incorporates both temperature and time. So you can see that this red line just represents 85 degree days. And you could get there several ways. If you're incubating these cysts at 10 degrees, it takes 8.5 days. If you're incubating them at 1 day, at 1 degree, it's going to take 85 days to get you. But you can see all of these different locations, including the Chukchi Sea, fall on that curve. So that's a really powerful tool. So how do we use it? Well, now we know the water's warmed 2.5, almost 3 degrees here. What does that mean? Well, basically, you can see up here, this is the temperature. This plot here is a factor change in germination flux. All it means is how many more cysts are going to germinate in a given season with this temperature change, at this temperature versus this temperature versus this temperature. And you can see that we are nearly a factor of 2 higher in terms of just the number of cysts emerging from the sediments with just a 2.5 degree warming, and a little bit less, but still an increase in the Barrow region as well. So, in fact, it's the other way around. You can see this one is higher than that, but still a significant increase. But maybe you think, what's the big deal? I know when I first saw the data, I told the people in the lab, I say, well, that's just one division. That's, 1 cell makes 2. That just gives you 1 more division, big deal. But then we started looking more at some of our own work in the Gulf of Maine, where we have established there's a direct relationship between cyst abundance and the extent, the geographic extent of the blooms in those regions. And there, a twofold increase in germination flux gives you about a 2.3-fold increase in the length of the coastline closed to shellfish harvesting. So if it was 150 kilometers of coastline closed one year, it could be 300 the next, with twice as many cysts germinating. And also a 6.5-fold increase in the cumulative shellfish toxicity we might measure along the coast. So it's far from trivial, but it's not the whole story, because warming temperatures have also expanded the environmental window during which these blooms can form. So what does that mean? So let's look at these figures. On the left, you can see 1 decade of temperature for this region with you can see the color, the warmer areas here. And that goes through 2008. On the right, it's the next decade, 2009 to 2018. And you can see that things have warmed. You can see very much warmer conditions up here in the Chukchi. And if you subtract the two, you get panel C here, where it shows you that anywhere, again, 2 to 4 degrees, but this also shows you where these temperature changes are occurring in these bottom waters. And if you look, we've outlined in this panel D over here, that this is roughly the cyst seedbed areas. And you can see they do include a number of these areas that have warmed a great deal. But if we take that function that I showed you about the number of days that it takes until germination, what you get then is this fourth panel, where this is basically the difference in the days until germination. And the areas that have this darker maroon or whatever color, brownish color, are between, let's say, 2 weeks and 3 weeks earlier germination. So with roughly 20 fewer days would be needed for a cyst to germinate in the Ledyard Bay region now than was a case in the prior decade. So we're not just getting the more cysts coming up, they're germinating earlier. And with doubling times at these temperatures for the surface water there, roughly 2.5 to 4.5 days for a cell to divide and double, you can see that with 2 to 3 weeks of time, you would have 2, 3, 4 more divisions. So the population could be much, much bigger with this head start. So this recent warming that we're seeing up there, we see it supporting not only earlier and on faster germination. I haven't shown you data for that, but it is true. Earlier and faster germination, longer periods that are favorable to the vegetative growth, with more rapid cell division and bloom development. And therefore, you've got this, whether double whammy, if you will, about that the warming is imposing on this region. All right, so back to the surface waters. I'm sure some of you have seen this figure, which was from 2019 in June, a very, very warm year, but it shows you, in some cases, the scale of the warming that's going on there. This is versus a 30-year average for this region. You can see that up in the area that we're most interested in, 3.5, 2.5, these are degrees increase over this long-term average. So things are much warmer in the surface waters. Well, what does that mean? Well, when this is another visualization of the bloom in 2018. And when we took that data, the surface water temperatures in the area of that bloom were roughly 8 to 10 degrees. And if you look at some growth curves that we've generated for isolates from the Gulf of Maine, and the Chukchi isolates are actually in red, and some from Greenland and some from Iceland, we see them all behaving very much the same. But if you look at this 8 to 10 bar here, it really just shows you that the Chukchi and these other isolates are growing 60, 70, 75% of their maximal rates at those temperatures. No problem at all for a bloom to develop. We typically don't see blooms in the Gulf of Maine ever reach these temperatures. They're always happening down here and blooming very, very successfully. So the waters are definitely supportive for vegetative cell growth. So that's sort of the data as we have it to this point. There's a number of things that are ongoing that I wanted to talk to you about. We are going to repeat the Alexandrium, and I'll show you a little bit more pseudonychia data and new pseudonychia data in a second. That's the domoic acid or ASP organism. We're going to using cruises of opportunity this year and next, and we have our dedicated NSF funded HAB cruise, which has been delayed multiple times. We don't know whether it'll be 2022 or 2023. If it is, this is where we'll go. We have coring studies and toxin and microsatellite analysis planned as well to determine the history. How long can we go back in time and see these organisms in the sediments? What's their origin of the blooms and the connectivity of the HAB populations in the region? And in terms of that connectivity, I'll just give you one example of what we're in the middle of doing. Alexandrium produces this toxin family called saxitoxin, and it's not just a single toxin, it's a suite of toxins, and we can separate those analytically, and they are in a sense a fingerprint for different isolates of Alexandrium. And that's what these little pie diagrams are showing you. This one up here is almost entirely producing saxitoxin with small little percentages of these other toxins. This one over here, in contrast, isn't producing any saxitoxin, but it's producing a lot of these C1 and C2 toxins. And as a bit of an aside, the reason this is important is that saxitoxin and a couple of these others are really potent toxins. The C toxins are really weak toxins. They're maybe 100 times weaker. So depending upon which populations are being brought into the system or which ones are resident there can have a huge difference on the amount of actual toxin being produced. But from a connectivity, a population connectivity standpoint, what we were trying to do and are trying to do is collect isolates from around the region. Now, these are data from many years ago that I produced with some Russian colleagues. And we have a little bit of data from that Swakey's paper and our own isolates. And what the intention is to see what is the distribution of toxin types and which ones are making it up into this region when we look at the sediments. Excuse me, but you can imagine it's difficult. If this is sort of the end deposit area, we should find up here a big diversity of toxin types. And we've got many, many isolates being analyzed now that we'll see whether that's true. But if we're there and able to collect during a cruise and actually collect cells during in the right way and isolate them so we get individual toxin profiles, we can start to separate which cells are in these bloom patches. And let's say we did it in 2019, which cells were dominating here and which cells were dominating here, and do they have the same toxin fingerprint? It would be yet another way to see what kind of connectivity there is. All right, so I said we're going to be doing these this type of work. We're also a new project funded from the NOAA EcoHab program with Kathy LaFay as the lead PI we're collecting and processing HAB samples from every possible aspect of the food web, from the cells to the forage fish, the zooplankton, the worms, the clams, everything we can see to try to look at the transfer of toxin through the food web. And obviously our goal is to try to understand and communicate the real human health and ecosystem threats from these HAB toxins, given that we know so little about these new vectors. Out in the Gulf of Maine and other parts of the world, people aren't eating the seabirds and the walrus or whales, and we just don't know these animals' lethal doses, and we don't know what tissues the toxins are accumulating in. There's so much to be learned here and then communicated to the communities. I said I'd talk to you briefly about pseudonychia. There's just one single slide. This is work being done by my collaborator, Kate Hubbard. And it's a complicated figure here, but it's also quite revealing. If you look at the map, these are the various transects from this one cruise. We've got more data, but this one I like because it's easier to understand. And you can see that these are the various DBO5, DBO3 stations, and they're down here. So this is a linear going from way down here, DBO2, all the way up to over here in the Beaufort area, in the Kenzie Delta. And what pseudonychia cells are very hard to differentiate from each other morphologically. So Kate uses a molecular method called ERISA that gives you a relative abundance. And what I want you to see is that the really toxic forms of pseudonychia, Pseudonychia australis, Pseudonychia pungens in red and green, and even this pinker one, which is Pseudonychia fraudulenta. These are, mildly toxic here, but the more toxic ones, the green and the red, are found right from the Bering Strait all the way up to DB05, and then they sort of disappear. And then you have a lot of this weakly toxic Pseudonychia delicatissima, and then quite interesting to me, a lot of these grays and blacks out here, which are just unknowns. They are Pseudonychia, but we don't know anything, what species they are. We don't know if they're toxic or not, and they're the ones occurring in these colder waters. So to wrap up, I hope I've shown you that there are multiple toxic HAB species in the Arctic, and I think they're likely to become more prevalent as warming temperatures and sea ice reduction expand the window in which these blooms can initiate and persist. But even though we're making some headway here, still very little is known about the origin, dynamics, and transport of HABs in these waters. So I've shown you today 2 extensive seed beds of this toxic dinoflagellate in the Alaskan Arctic. Bottom temperatures are certainly high enough to support in situ germination. Now they are. High concentrations of planktonic or vegetative cells were also detected both years, and again in 2020, but again with temperatures conducive for vegetative cell growth. Analysis of the warming trends that I showed you said that we've increased the germination flux approximately 2-fold during the past 2 decades, and we've also advanced the onset of bloom initiation by about 20 days. Together, all of the data suggests that we can have blooms that originate and grow locally, and therefore they're likely to be what we call self-seeding. They grow, they initiate, they bloom, and they drop new cysts, like I showed you in the Gulf of Maine, and therefore you get these recurrent outbreaks. And evidence also suggests that southern populations transported through the Bering Strait may be a second source of blooms north of the strait. So I started out with the Gulf of Maine. I'm saying the situation has some real similarities. Well, let's look at that real quickly. If we look at the 2, what do we have? Causative organism is the same. 2 major cyst seedbeds, the same. A coastal current covering hundreds of kilometers in the Arctic from the south to the north, and the Gulf of Maine from north to south. And the region is impacted by both advected blooms and localized self-initiating blooms, the same. Excuse me, but then things start to deviate a little bit. The cyst seedbed in the Gulf of Maine is large, I can say now in retrospect. The one in the Arctic is massive. In the Gulf of Maine, bottom and surface temperatures have long been supportive for in situ germination and vegetative cell growth. And I believe in temperatures in bottom and surface waters are only recently emerging from inhibitory levels in the Alaskan Arctic. So in the Gulf of Maine, recurrent large-scale regional blooms have been occurring for decades. And here we think that recurrent large-scale regional blooms were probably sporadic in the past, but are more common now, and this seems to be more of a recent phenomenon. In terms of food safety, it's a well understood threat in the Gulf of Maine. We understand shellfish, how to protect people and resources, but it's a very poorly understood threat to food safety in the Alaskan region. And there are effective monitoring programs in the Gulf of Maine, but essentially no routine monitoring programs for recreational or subsistence shellfish harvesting, and certainly nothing right now for other subsistence foods. So to continue the summary, the trail of death hypothesis, as appealing as it is, does not appear to be valid for the present conditions. It may have been true in previous decades. So the sleeping giant may have woken up. All of these cysts that we were saying were there waiting for it to warm up. Well, it's warmed up and they're waking up. The multiple Pseudonychia species were observed throughout these cruises, some of which are highly toxic. And so HABs clearly have significant implication for Arctic food web dynamics and can affect public health as well as vulnerable marine bird and mammal populations. But I want to make sure everybody gets this point. Everything here that I've said so far talks about the potential for significant and increasing HAB impacts on human and ecosystem health. But thus far, there have been no human illnesses or deaths associated with these blooms or toxins in recent years that we know of. So we're still trying, there's a threat, there's no question, we have to study it, we have to understand it more, but this is not something for people to get overly alarmed at, because so far people are not getting sick. So with that, I'm going to finish. Certainly want to acknowledge funding from the NOAA, GOMO and ARP program, NOAA EcoHab and NSF polar programs, along with all of these people who have helped with so much of the collection. So with that, I'll stop and I'm happy to take questions.
Thank you, Don. That was a really great presentation. I really appreciate that. We are now going to go into the Q&A session, and there are already several great questions in the chat that I'll get to. Just a reminder to everybody, if you're joining by Zoom, please type your questions into the chat window. I'll record them and read them aloud. If you're joining by phone, please raise your hand by pressing *9 and unmute yourself by pressing *6 to ask your question aloud. Dr. Anderson has offered to continue to be available for another 5 to 10 minutes after the top of the hour so that we can continue this conversation. So I'll get right into the Q&A. Dave Checkley asks, do genetics offer insight into what the bottom pool surface blooms originate from?
Okay, thanks, Dave. The answer is they should, but it takes some time. The method that we are using, first of all, to look at the connectivity between regions involves taking cyst samples that are the easiest to work with. And we need to isolate, to do the microsatellite analysis, we need to isolate on the order of 30 or 40 cysts and germinate them and get a culture that we can extract and analyze for microsatellites. And we have to do that at a number of locations along the coast, and that can help us understand connectivity. If you can imagine doing what you suggest, which is staying in an area and isolating, well, first of all, it's easy with the cyst population. You could save that sediment, bring it back to the lab and isolate it. But we then need to look at the bloom population and on board the ship, isolate in a number of different locations, 30 or 40 individual cells with micropipettes and such, get them into individual cultures on the ship with incubators and so forth, with enough time for them to grow out before we go back to port, so that each one of those, so that you get one part of one bloom, maybe you get 30 cells, you move to another place, another 30 cells, another 30 cells. It's something that we are talking about doing in the course of a cruise, but we're going to have one very hardy person doing that with all of these single cell isolations on a moving ship for 20, 25 days or something. So yes, the answer is yes, it should tell us some good information, and we have hopes to do it, but it hasn't been done yet.
Thank you. Next question is from Robert English. He says, thanks, Dr. Anderson. I understand factors such as current velocity and temperature gradients that correlate with cyst deposition and germination in particular regions. Are there also subsea geologic features such as mountains, canyons, etc., that might be part of the explanation too? Or perhaps different types of silt sediment on the seabed that either help cysts accumulate, persist, and eventually germinate?
Great. Well, I don't know about features that go way down deep other than their effect on just general bathymetry. If you have a deeper basin, for example, that tends to accumulate fine-grained materials and that reflects some sub-bottom structure, then yes, there would be a correlation. But I think the second part of your question is where the answer lies. The cysts tend to behave like the finest clay and silt particles. So they will, if you drop a cyst in a really sandy area, that's a high energy environment, they won't remain there long. They will get resuspended and advected, and where they ultimately end up will be in these depositional zones. And that's typically in other parts of the world than where you have the highest concentrations of silt and clay. And so that we are working, Jackie Grebmeier's been working forever in that region and has a wonderful knowledge of the bottom types and the communities there. And I know she's told me at times that some of our cyst hotspots are not located in her benthic hotspots. And so I think we need to sort out that possible discrepancy. But typically, yes, these cysts accumulate in areas where the velocities drop and where there's deposition zones where resuspension is more difficult and you get accumulations.
Thank you. Thomas Ferrugia writes, fantastic talk, Don. Thank you. Have you read the recent UNESCO report on global HABs over time? It doesn't seem to show a consistent pattern of increased HAB events with changing ocean conditions. What was your reaction to those findings, and how does your work in the Arctic fit into that global picture?
Well, the answer, Thomas, is I'm actually an author, a co-author of that paper that just came out in one of the Nature publications. And I obviously agree with the findings, but they need some explanation. The finding is that that was an effort to look globally at all HABs of all types over all countries where we have been collecting data for the last 30 years and more. And if you look at that with the proper statistical approach, you find there is no global increase of all HABs. And that's not surprising, but in fact, you probably didn't catch it. In one of my first slides, I talked about the problem is increasing in frequency and magnitude, and I added in some regions, because that's the other finding of that Nature paper, is that globally, if you lump everything together, there's no common trend. But it makes sense. These are different oceans, different climates, different forcings. Some are polluted, some are not, some are pristine. It doesn't really make sense that you'd expect some major trend. But if you look in individual regions, you definitely do see trends. I wrote a US companion paper to that Nature paper where we looked just at the US, and where we don't see an increase in certain syndromes like PSP or DSP or others, we do see increases in some of the other syndromes. And we also do see a national increase in HABs that is statistically significant if I pool them all together. So it's all regionally based. And so what I'd say about Alaska is that we need to look at Alaska in isolation and try to get the data that we can use over time to detect trends. And that, unfortunately, that's going to take decades to be able to really see these trends. But I do believe that if we can get it going now, that we will see some remarkable trends, and they may include increases in these HABs that we're looking at.
Thank you. Nadia Steiner asks, what depth level do the vegetative moving cells occupy, i.e. where do we have to look to estimate further distribution into the Arctic?
Well, actually, maybe I should, let's see, let me go back. I don't think I'm sharing my screen anymore. I had an extra slide here. So this just shows you along that one of the Ledyard Bay transects in 2018. To me, this was a rather remarkable figure. On the right, it's onshore, and on the left, it's offshore. And you can see the yellower colors are high cell densities, 5,000 cells per liter. And you can see them distributed in at least the top 30 meters of the water column here, and tapering off as you go further offshore. But even well offshore, there are a reasonable number of cells down 10, 20, 30 meters. So the answer is that vegetative cells are largely going to be in your mixed layer, and however deep that is. If you get some good mixing that takes them top to bottom, then that's where they're going to be. And then the Chukchi shelf is relatively shallow, and it wouldn't surprise me at all to find vegetative cells down near the bottom. Likewise, I wouldn't be surprised, and we have in fact seen cysts up near the surface that were probably resuspended. So there, and part of the reason this was a remarkable slide to me is if you take this kind of abundance and over these depths, and you look at the size of the bloom that you see here, it's an extraordinary amount of toxin. It's covering 100 or more kilometers and 40 or 50 kilometers offshore and 30 meters deep. There's a lot of toxin there. And so where's it going and what's it affecting?
Thank you. We have a few other questions online, but I see it's near the top of the hour, so I'm going to do a soft close, and then Dr. Anderson has agreed to stay on for another 5 to 10 minutes to continue discussion. For those of you who are leaving now, we want to thank you for joining us today and extend a special thanks to our speaker, Dr. Anderson. Today's webinar will be archived and it'll be available at a later date at www.arcus.org Research Seminar Series/archive. You can also visit the Arcus homepage for more information about Arcus, our programs, and upcoming Arctic-related events. We are always looking for ways to improve our seminar series and also for suggestions for future seminar topics and speakers. Please do take a minute or two to complete our short survey. A link to that is available now in the chat as well as on the window on the screen. The survey will also be sent to everyone that's participant as a follow-up email once the seminar has been posted. Let's go back to the questions then for those that are able to stay online. The next question is from Andrea Snyder. She says, thanks for a great presentation about the high number of cells in the Alaska sediments and trails of death hypothesis. How old are the sediments that hold the cysts? This may give an idea over how long the cysts are accumulating there and puts this in context with climate and surface water warming.
All right, well, one of the intentions of this often postponed NSF cruise was to do some dating of sediments in the area of the cyst high abundances, and to also look at the cysts or even the ancient DNA for Alexandrium and to try to see how far it goes back. But we have not done that yet. I know Jackie Grebmeyer was on the talk. I don't know whether she is there or can help answer that question, but I know that it's difficult to do dating in some of these sediments because of the distance from, there's not a lot of lead-210, I guess, signature there. So I really don't have a good answer at this point. We've only just started to try to address that, and because we haven't had our crews to get our own data, I haven't delved into it very much. So if someone else knows, maybe they can put it in the chat, but sorry, I just can't answer that one.
Thanks, great suggestion to include more discussion in the chat. The next question is from Jackie Clement Kinney, and she says, excellent talk. I'm wondering, what is your speculation on populations of Alexandrium catenella in the Western Chukchi and Eastern Siberian Sea? In the model I use, which is the RASM, we see a strong flow through the long strait and high primary production in the ESS, as well as the Chukchi.
All right, that's a very timely question, and thank you for asking, because I forgot to mention one thing, which is that it's obvious that we need to know more about what is happening to the west of the boundary where we can't sample anymore. And the good news is that we recently were notified we have funding from this Beringia, I guess it's a foundation, or it's an organization that combines workers from the US and Russia for work in the Bering Sea region. And we will be collaborating with Russian HAB colleagues to try to answer some of these questions. They can't send us sediments. We are teaching them to process them the way we do them, so we can then start to help them choose locations, and we can expand our cyst maps using identical methods where possible. We're also going to be making isolates so we can do some of these connectivity studies with microsatellites and toxin composition. So we will be starting to get data in the next few years from the area you're talking about in the Western Chukchi and the Eastern Siberian Sea. But right now we have nothing. If you ask for my speculation, I think we are almost surely going to see populations there. Certainly an extension of both the cyst map and the vegetative cell map suggests that those features extend off to the west. How much they impact the coast, I don't know. There is a coastal current, a Siberian coastal current that flows to the east, and maybe that makes it harder for populations that are offshore in the Western Chukchi to reach the communities on that coast. I don't know. I don't know anything about that, the oceanography of that region, but it's something that we will try to be addressing through direct collaboration with these Russian colleagues. And that, I should mention that also, that project includes a heavy element of community outreach. So we should be working with local communities on the Russian side as well as on the Alaskan side to learn what we can from them about risks and so forth, but also to communicate what we know.
Thank you. Jennifer Francis writes, the Bering Sea areas has been warming even faster than the Chukchi Beaufort region. Are there any data from there, or do you have any plans to go acquire some?
Yeah, thanks, Jen. She certainly would know. She works hard in this type of field, but the answer is no. There is no data from there, and I know my colleagues at the Avi have plans to do more cruises looking in that type of region, but I haven't heard anything recently. And right now, our hands are full, as you can see, in this Alaskan Arctic area. So maybe we'll get a chance to join one of their cruises again. They've been great at inviting us in the past, but that's a good bit of motivation to look hard there. But you need you need a few things to make it. It's not just the warmth you need there. You need a source of cells, and as I told you, that whole if you look along the coast of Greenland, the numbers are low, and I don't know how. It's not like the Bering Sea where you can have, or the Gulf of Alaska where you can have these huge dense plumes that can then be transported up and into a region. I'm not sure where the source would be for that area, but it's certainly an interesting thing to look for.
Thank you. Judy Fonstock shares that to learn more about Dr. Anderson and his team's research in 2022, we hope, please follow the Polar Trek expedition that he's running with teacher Eric Filiardi. And she has a link to that in the chat, which is polartrek.com, expeditions harmful algal blooms in the Arctic waters. And then several kudos coming to you from different people that are on the chat, including Jody Payton, thanks very much. This is fascinating. Jennifer Francis writes just now, thanks so much, Don. It's exciting to hear about your latest adventures. CV Anhert, I believe is pronounced, says, thanks, great talk. Kristen Beam, thanks and excellent. So I think that unless there are more questions coming up, Don Anderson, do you have anything else you'd like to add before we close the webinar?
Well, I guess I want to emphasize that last point I made. When I was asked if I would be willing to post this talk on the ARCIS site, I said eventually, but right now we have a publication that's been well reviewed, and we just need to go with some minor revisions. But when it comes out, I wouldn't doubt that it's going to attract quite a bit of media attention. And you will hear me and others take this cautious approach, but we need everybody to take that approach when you talk with people. If this subject comes up, that we walk a fine line in our field of harmful algae. We need to describe the dangers that are there. And there's no question there's toxins. I've showed you there's a lot of toxin. I've showed you why I think it's going to be happening for many years to come. But how big a risk is it? We don't know yet. People don't eat a lot of clams up in the North Slope. They collect them when storms wash them in, and we know that some of those deep water clams that have been washed, the big ones that get washed in, have had dangerous levels of toxins in them. But we just don't know just what kind of threat there is there. So, like I say, it's a fine line for us to walk and a fine line for all of you. But the message is there's something to be concerned about. We're on it. We're studying it. Lots of agencies are doing measurements, and we're talking to each other. We're communicating, and we're trying to define the risk, but we don't want it to be over sensationalized.
Thank you. Well, with that, I think we're ready to close the webinar. There continues to be great kudos coming to you in the chat from a lot of different people. And with that, on behalf of ARCIS, I want to thank you, Dr. Anderson, for a really fabulous talk today.
Okay, my pleasure.
Yeah, thank you. And as he said today, well, today's webinar will be archived and available when that paper is, when it's public. But in the meantime, you can visit the Arcus homepage for more information about Arcus and our programs and upcoming Arctic related events. And we will send a link to the archive when that's available to everyone that's registered. We're always looking for ways to improve our seminar series again, and looking for suggestions for future seminar topics and speakers. So please do take a minute or two to complete our short survey that is available now and again in the chat window. And a survey link will be provided to you in a follow-up email once our seminar is posted. I want to thank all of the participants today and our speaker, and I encourage you to continue to stay engaged with us. Thank you again, and wishing you a very good day. With this, our seminar is concluded. Thank you, Don.
Yeah, bye.
Bye-bye.